Today I'm going to talk about the central question in sleep, which is that sleep continuity is essential for its function. And there's plenty of evidence supporting this statement. When we don't have a continuous night of sleep, we feel really bad, and animals also feel really bad, and they forget things. But we don't know really much. We don't have much detail about the mechanism providing sleep continuity. And central to sleep continuity is the role of these neurons in the lateral hypothalamus that we identified 20 years ago, that we call hypocretin, also known as orexin. And these neurons are localized in the lateral hypothalamus, only a few thousand of them, that innervate the entire brain. So why are these neurons, or why I say those neurons are central? That's because work by many labs, Masashi, Yanagisawa, Emanuel, and others, have shown that when you are missing these neurons, you have narcolepsy, which is a disruption of sleep architecture. And many of you know about this, so I'm not going to get into the details. But these neurons, or the discovery of these neurons, has led to important advances in the sleep field, including the development of drugs that target the hypocretin receptors. Suvorexant is a non-selective hypocretin receptor antagonist that is approved by the FDA for insomnia. And the crystal structure of the hypocretin orexin receptors was determined a few years ago and has allowed the development of selective agonists that eventually will be used to treat narcolepsy. So one of the questions that we've been trying to address is how do these neurons affect sleep continuity? And in particular, what is the algorithm? What's the architecture? What's the logic behind these neurons stabilizing sleep? So when we talk about computations, we went to the most basic computation of all, which is the perceptron. And the perceptron is an algorithm shown here. Very simply, inputs are assigned weights, and those are fed into a net input function that sums or integrates these inputs, and then that function is fed into an activation function that decides in a very simple operation. It's a step function, so it decides a binary. It makes a binary decision, yes or no, and that's the output. So a sleep perceptron would look something like this, and where we have multiple inputs assigned different weights, integrated into this function, and those are the outputs. The outputs are the arousal monoaminergic nuclei. And what's the nature of these inputs? The nature of these inputs is very broad. It includes food, metabolic state, circadian rhythms, as we've seen throughout the day. Positive and negative emotions. We know that availability of food and a sex partner inhibits sleep. Negative emotion, stress, also inhibits sleep. And of course, sleep homeostasis and history and so forth. So the question is, how do the hypocretin neurons integrate all of this information? And we've been accumulating evidence over the years that indeed those neurons integrate all sorts of information, including of the types that I've shown you. And evidence for that is, like I said, very broad. I'm going to summarize this very briefly. The hypocretin neurons as a whole express a whole bunch of receptors, GPCRs, that are sensitive to, that actually communicate these variables to the hypocretin neurons directly. But also there's additional information conveyed to hypocretin cells through local circuit neurons in the lateral hypothalamus. Some of these include the leptin receptor positive cells, that are, by definition, sensitive to leptin, which is a hormone that is released by the gut and provides information about the general metabolic state. So these leptin receptor positive cells are present in the hypothalamus, here in green, and they're intermingled with hypocretin cells. And we demonstrated a few years ago that indeed leptin receptor positive cells modulate very efficiently hypocretin neurons, and therefore hypocretin neurons, without having leptin receptors, they're modulated by leptin. So more generally, the long-range afferents to hypocretin also modulate their activity, and that is more challenging, but we've been addressing this more recently. So we've done a map of the monosynaptic inputs to hypocretin neurons throughout the brain, and these are just a few examples. The histaminergic cells in the tumeromammary nucleus, and the bed nucleus of the stria terminalis, the central amygdala, and so many others. So, and I'm going to give you just examples. I mean, these are just snippets of information of how the bed nucleus of the stria terminalis projects to hypocretin cells. And actually, there are many types of BNST neurons that project to hypocretin. And Will Giardino in my lab identified 2 of them that can be labeled with 2 different markers, CRF and CCK. And both project to hypocretin. What's interesting is that CRF neurons in the BNST are sensitive to aversive stimuli. So this is a photometry trace of calcium activity in these CRF BNST neurons. So they're sensitive to predator scent, but not to rewarding stimuli. And stimulation of these neurons is aversive. Okay. In contrast, the CCK neurons in the BNST that also project to hypocretin have the opposite profile of excitation. So they're sensitive to rewarding stimuli, in this case female scent, and not to aversive stimuli. And stimulation of these CCK neurons is rewarding. So again, we have evidence for inputs to these hypocretin neurons of neurons and stimuli in all of these categories, but I'm just providing a few examples of these. And like I said, the outputs of these neurons include arousal nuclei, noradrenergic locus ceruleus, dopaminergic VTA, and histaminergic cells. And we've also shown over the years that each of these monoaminergic cells individually is sufficient to induce sleep-to-wake transitions. So a signal coming from hypocretin neurons that activates any of these would eventually elicit an arousal response. And this is work already published many years ago, showing that indeed locus ceruleus activity is sufficient to induce an awakening. This is one of the first optogenetic experiments. So sleep disruption, like I said in the initial statement, affects sleep function. And we showed, actually, Asya Rolf, who is now at the Technion in Israel, showed that when we disrupted sleep optogenetically, essentially we fragmented sleep optogenetically, that affected memory consolidation. So this was important because it was not, it showed that it was not sleep loss that affected memory consolidation, but just fragmentation of sleep was sufficient to disrupt memory. So we asked the question: Is this a problem in the aged brain? So we all know that sleep quality decreases with age, and the question is, well, if sleep fragmentation, which is one of the features of of sleep during in aged animals and humans. If sleep is fragmented, will that cause deficits in cognitive function in and of itself? So, and we also asked, why is this that sleep quality and sleep fragmentation increases with age? So the hypothesis was that the arousal circuits that we have identified over the years are dysfunctional during aging. And we had some evidence for that because when we used fiber photometry, which directly assesses neuronal activity of individual neuronal circuits, when we looked at the activity of hypocretin neurons during sleep-to-wake transitions in young and aged mice, we saw a very clear decrease in the amplitude of the response in aged animals. Okay, and this could be due to that the decreased signal could be due to many possible scenarios. One of them was that we had actually cell loss of hypocretins, and indeed this shows that the aged animals have a dramatic reduction in the amount of hypocretin neurons, about 20 to 30% reduction, which is much more profound than neighboring cells. So we have fewer hypocretin neurons in aged animals. Are they impaired at eliciting sleep-to-wake transitions? So we looked at that by using optogenetics, as we've done in the past 15 years. And we looked at optogenetic stimulation of hypocretin neurons in the lateral hypothalamus in young and aged animals. And what we observed was that in aged animals could elicit sleep-to-wake transitions very, very efficiently. But the length of the wake bout in aged animals was much, much longer than in young animals. So essentially, there were fewer hypocretin neurons, but they were very efficient at inducing sleep-to-wake transitions. And something in the circuit was affected so that a single stimulus evoked a much longer response. So there were several possible scenarios for this effect. One was that the cellular environment is getting old, and therefore some mechanisms of cellular senescence are taking place. Also, that aging causes an increased synaptic input to hypocretin neurons. That is a possibility that has been described in other areas of the brain. And that aging affects intrinsic excitability of hypocretin neurons. And we explored each of these options. So we looked at single-cell RNA-seq of young and aged neurons to look for markers of cellular senescence, and we didn't succeed. So essentially, we saw that the mRNA profile of aged neurons was exactly the same as young neurons. Only a very few mRNAs showed a difference, including hypocretin, but there were no signs of cellular senescence, in short. So Gordon Wang, who works with Philippe, helped us with identifying markers of the cellular environment in the hypothalamus, and he looked at the synaptic inputs directly to hypocretin cells in young and aged mice. And indeed, one of the hypotheses was true, that aged animals have many more synaptic inputs, excitatory synaptic inputs, compared to young mice. Why is that? We don't know yet. And we also explored the possibility that the aging increased the intrinsic properties of hypocretin cells. And so we recorded these cells in slices, young and aged mice. This is not easy, but took a while, but we succeeded finally. And this is what we found. This is the proportion of spontaneously active neurons in young mice compared to aged mice. You see that there are many more spontaneously active neurons in aged mice compared to young mice. So they're hyperexcitable somehow. And indeed, recording individual neurons told us that the resting membrane potential of aged animals, of aged neurons, was much closer to threshold. So they were indeed depolarized. And the response to a single stimulation was very different in young versus aged. You see this optogenetic stimulation of a young hypercretin neuron. You see a single action potential, where the same stimulation of an aged neuron shows this jittery response. So what could cause this change in intrinsic excitability? Well, a few years back, we generated an in silico model of a hypercretin neuron using these Hodgkin differential equations. And so this is an in silico trace of a modeled hypercretin neuron, okay, that very much looks like an in vivo trace. So we asked our collaborators to modify each of these components of the equation and see which was most important in changing the excitability of the hypercretin cells. And when we changed the potassium conductances, this is what we observed. That cells were much more active, much closer to threshold, easier to depolarize. So we hypothesized that changes in potassium channels could account for this hyperexcitability in aged neurons. And indeed, that's what we proved. So these are young neurons treated with a potassium channel blocker, increase the excitability, and this very much looks like an aged firing profile. And conversely, this is the aged, the trace of an aged hypercretin neuron. When we treat these slices with a potassium channel opener, we sort of rejuvenate their firing profile. And this also happens in vivo. So when we treat young animals with potassium channel blockers, we disrupt sleep. And treating aged animals with potassium channel openers rejuvenates their sleep and consolidates, reducing fragmentation. So in collaboration with Hannah, who's here, I think, and NASA, Synod Armstrong, they looked at possible correlates of these potassium channel expression in humans, and they found indeed that there's a significant decrease of potassium channel expression in postmortem tissue in humans with these channels. So this could also apply to aging in humans. I'd like to acknowledge the people who did the work. So Will Giardino did the BNST work and shipping all of the aging work, and this is in collaboration with Li Chun Lo, Hannah, and Gordon, and Greg Scherer. Thank you.
Thank you for being perfectly on time. Questions?
No questions.
I have a question about your pharmacological study. Maybe I missed them. When you give blockers of potassium channels, etc., how do you know that it's specific for the hypocretin I guess my question is, do you know whether there is an indication that this increase in excitability could apply also to other arousal systems or other cells?
Right. There's nothing specific to hypercretin in our manipulation, but I've skipped a couple of slides showing that pharmacological treatment affects. So we recorded hypercretin neurons after pharmacological treatment, and so hypercretin neurons are affected, and so are many others. But obviously, those neurons are critical to drive sleep-to-wake transition. So if you affect their excitability, it's going to have a major effect on sleep architecture, period.
What about the cortex, for instance, something not directly or not so directly, at least through the arousal system? Do you know if there is changes in excitability?
There are, but in the opposite direction. In the opposite direction, yeah.